Hi everyone, I'm Loan Vulia from the group of Jörn Meimke at the Max Poets Labs and the Center for Molecular Medicine in Vienna, Austria. And today I want to introduce BioProfiling.jl, our package dedicated to the analysis of high-content imaging screens. These screens usually follow the same set of steps, starting with a library of perturbations. That can be, for instance, a drug library or a guide RNA library for CRISPR screens. And they're used on cells in an array format, in microplates, and in each well of these plates, we are acquiring fluorescence images on which we can measure a lot of different parameters describing the shape of the cells, as well as the intensity and distribution of the fluorescent dyes. This information can be compiled into quantitative profiles describing the state of the cells in each experimental condition. And these profiles can then be used for different applications. For instance, you might be interested in identifying morphological clusters, or you might want to try to predict the mechanism of action of candidate drugs. So today we're interested in going from images to knowledge. And in the case of high-content imaging screens, this can be quite a challenge. Starting from the images exported from the microscopes, if we have image stacks, we would first project them and correct the illumination. Then we can segment different objects, such as the cell body and the nuclei, and we can perform measurements on such objects. There are multiple ways to do that. For instance, many people like to use cell profiler or deep learning techniques, and there's room for improvement, but there's a lot of choice available already. And today I want to concentrate on what comes next. Once we have these measurements, we typically like to filter out imaging artifacts, aggregate profiles per condition, and normalize the data and select informative and non-redundant features so that we end up with profiles that are summing up the cellular morphology in each experimental condition that are comparable and usable in downstream tasks. One of the biggest challenges we have to face in high-content imaging screens is that imaging artifacts are really common. And for instance, we often observe precipitations or clots of the fluorescent dyes or other types of artifacts. So to make it more quantitative, I went over example images from a few good screens and checked by eye how often such artifacts were observed. That's what I'm showing you here. So each column is one study, and the colors correspond to different types of artifacts. And the part left in brown corresponds to artifact-free images. And this tends to be uncommon in some screens. So this is happening really with even the best experimental setups, and does not always mean that we have to completely discard the images. But the key message here is that we always end up with some contamination in our data and that we must deal with it somehow. And that's precisely why we have developed BioProfiling.jl. This is a Julia package that comes with dedicated data structures for morphological profiling, tools for visual diagnostics, tools for quantifying the distances between morphological profiles, and ways to interpret what changes in morphology means. So I will briefly tell you about it, and if you're interested, we have a paper out there already, which is available in open access, so please check it out if you want more details. This is a Julia package, and you might not be familiar with this programming language yet, but we chose it because it's extremely fast and quite convenient. Here I'm just showing you some benchmark data compared to other programming languages, and different tasks are shown with different colors, and the more on the left you are, the faster you are, and Julia is consistently among the fastest programming languages, even comparable to C, but the syntax is much closer to Python or R and much more user-friendly. And if you're curious, you can find more reasons to use Julia here. Now I will illustrate what BioProfiling.jl can do. And for that, we conducted a drug screen where we tested a few hundred different drugs on U2S cells. and observed how the cells look like before and after treatment. Here is an example. DMSO is our negative control, and vinblastine is a tubulin inhibitor, and you can see that the cells that are not dying have a really distinctive morphology. So what we did is that we processed all of these images and measured descriptors of the morphology of the cells using Cell Profiler. And these measurements are what we used as input to BioProfiling.jl. After removing obvious imaging artifacts, we aggregated our measurements per image and removed redundant features, transformed our data, and then we were ready to visualize it. BioProfiling.jl makes it easy to visualize profiles, and here we see with a UMAP plot that some compounds, such as vinblastine again, are affecting the morphology in specific manners, and consequently we see that they correspond to different clusters in this plot. I also obtained the measurements that were varying the most in the drug perturbations compared to DMSO, thanks to a one-liner in our package. And here, let's not worry about the details. I just wanted to point out that we have measurements linked both to the cytoplasm and to the nucleus, and to all three channels that we imaged that were relevant. So really all aspects that we investigated in this experiment were compiled and useful in the profiles that we curated. Our package offers ways to quantify the distance between profiles, and as such, we can detect hits by comparing the effects of each drug to our negative control. We implemented statistical tests based on distances that are robust to outliers to avoid biases induced by imaging and segmentation artifacts. And here I'm showing you what happens when we look at the robust Hellinger distance between images for each drug compared to our DMSO images. We find that many compounds, so the one on the left of the dotted line, are inducing significant morphological changes. Then we can look at what these morphological hits are, and for instance, we can see that all the tubulin inhibitors that were present in our screen, so as they are affecting directly the cytoskeleton, they were found to be morphologically active. And on the other hand, some other drugs, such as topoisomerase inhibitors, they didn't consistently change the morphology of the cells. We can even contextualize all of that by comparing our morphological profiles to other information that we have about the drugs. And here, what I'm showing on the right is a comparison of the similarity between the morphology induced by the drugs and the closeness of the targets on a protein-protein interaction network. So I just wanted to point out that there is some information that is overlapping between the different ways to look at our drugs, but the morphology in itself is bringing quite some complementary view of drug effectiveness. And what's next for BioProfiling.jl? So we are not specifically a tool development lab, but we are as well interested in applications. And we are right now using BioProfiling.jl to analyze some chemical genetic screens. And we are as well not giving up on our tool, but we are maintaining it and adding even more features, trying to make it compatible with other packages in the Julia environment. And we are as well planning to implement features requested by users. And if you are now curious about BioProfiling.jl and give it a try, please let us know if there are things that you think would be nice additions, and we'll see what can be done. And with that, I want to thank the organizers for giving me the opportunity to give this talk to you today. And I want to thank you for your attention, and I would be happy to answer your questions.